Good morning. I like to wake up with a cup of coffee and read the newspaper. Everyone knows that caffeine is a stimulant, but have you ever wondered why you can drink it, and in fact quite a lot of it, and not have seizures? On the other hand, if you drank even a small amount of any number of other stimulants like strychnine, you wouldn't experience wakefulness, but seizures and convulsions. Today, we'll be discussing stimulant compounds, some drugs you may be familiar with already, and hopefully we'll integrate that with new information that'll be useful when you analyze drugs that are developed over the course of your career. These four examples also provide a basis for extrapolating to the diagnosis and treatment of related but perhaps unidentified poisons that may act the same way. So stimulants are basically compounds that can activate a particular area of the brain. We're going to compare and contrast four common stimulants to see how they differ from each other in the brain and in the effects they produce. Some are toxic, even lethal, and others are clinically important drugs used to stimulate respiration. First, let's see where these drugs act based on your knowledge of neuroanatomy. Starting at the top of the brain, methylxanthines act primarily in the cortex to produce a mild stimulatory effect. This category of drug includes compounds like caffeine, theophylline, and theobromine. The reticular activating system in the brainstem, the area involved in wakefulness, is very sensitive to the stimulant effect of cocaine and amphetamines. While these drugs are not clinically useful, stimulants in this category are potential toxins for pets. The respiratory center, located in the medulla, is the primary site affected by the drug doxapram, or otherwise known as Dopram-V. For this reason, this compound is useful because it can be used clinically as a respiratory stimulant. Finally, the spinal cord is the primary site of action of strychnine, a rodenticide. While it may not account for many admissions to your clinic, strychnine causes a characteristic seizure that's easily identified. It's important to know how to identify and treat it because if left untreated, it's usually fatal. The methylxanthines are found in tea in the form of theophylline and in chocolate in the form of theobromine. Caffeine is widely distributed in coffee, tea, sodas, cocoa, and chocolate. You're probably familiar with its mildly stimulant properties. But how might you encounter methylxanthines in the clinic other than your beverage of choice each morning? In addition to being in tea, theophylline is the main ingredient of pills and sprays that are used as bronchodilators. These preparations are especially useful in dogs, cats, and horses. Another way that methylxanthines become important clinically is that chocolate containing theobromine is a common toxin, especially over the holidays when treats are left out all over the house. Even throughout the year, several over-the-counter drugs contain lots of caffeine. For example, Excedrin, Anacin, Midol. In addition, caffeine can be purchased in concentrated forms, such as no-dose tablets. When theophylline is used as a bronchodilator, it can either be in the form of theophylline itself, or because it has poor solubility, it can be combined with ethylene diamine to make it dissolve more readily and allow higher concentrations. In that form, it's usually referred to as aminophylline. These compounds are used, for example, in cases of feline asthma, congestive heart failure, or pulmonary edema, just to name a few of the most common applications. As you can see from this, asthma tightens the bronchiolar smooth muscle, it causes inflammation of the lining and production of a huge amount of mucus. Theophylline, available in a variety of trade names, blocks these effects. It also blocks inflammation supported by the infiltrating mast cells. Sodium cromolyn, a mast cell stabilizer, and glucocorticoids, an anti-inflammatory compound, are also used for asthma. So how do methylxanthines work? And why is chocolate a toxin, whereas theophylline is anti-asthmatic? We'll talk about their effects in the order of their sensitivity. At the lowest doses, methylxanthines antagonize adenosine receptors. Many of these receptors in the brain are inhibitory. Activating them by the release of adenosine contributes to the induction of normal sleep. So blocking the action of adenosine by inhibiting the adenosine receptors allows the brain to be slightly more excitable. There's a ceiling to this effect because adenosine only suppresses wakefulness, generally in the range of normal. Other adenosine receptors are excitable, and blocking them causes just the opposite effect. At higher doses, methylxanthines also inhibit the enzyme phosphodiesterase. This enzymatic action degrades cyclic AMP. As a result, inhibition of phosphodiesterase results in an elevated concentration of cyclic AMP in the brain and throughout the body in response to G-coupled receptors linked to cyclic AMP. Receptors that generate cyclic AMP tend to cause excitatory effects. Methylxanthines also compete with benzodiazepine sites at very high doses. This probably doesn't contribute much to its mild stimulant effect, but it may contribute to its toxicity. At even higher concentrations, methylxanthines inhibit the uptake and storage of calcium in the sarcoplasmic reticulum of muscle cells, or myocytes. Sarcoplasmic reticulum is a specialized smooth endoplasmic reticulum localized in the muscle. Instead of synthesizing molecules as the endoplasmic reticulum does in other tissues, in the skeletal muscle, the sarcoplasmic reticulum sequesters calcium to terminate the shortening, or that is the activation, of the muscle fibers. When calcium remains outside this organelle, the muscle remains excited longer than usual. But it takes a lot of caffeine for this effect to develop, so it's generally in play only during toxicity. The first symptoms you might see in a case of methylxanthine toxicity would involve adenosine receptors that are sensitive to low doses of these drugs. There are 4 known subtypes of adenosine receptors: A1, A2A, A2B, and A3. They're all G protein coupled sites. A1 and A3 sites are inhibitory, such as those that induce sleep, and A2B and A2A are excitatory. As you can see, they either inhibit or stimulate adenylate cyclase activity, which then influences calcium and potassium transport across the membrane. Adenosine receptors are located in many areas of the body, not just the brain. When adenosine is released, in addition to sleep, it may be involved in epilepsy, migraines, stroke, pain, respiration, and inflammation throughout the whole body. Blockade of these receptors at specific adenosine receptor subtypes is an ongoing subject of investigation into new ways to treat a variety of conditions. The major use of theophylline is for asthma, and the major use of adenosine itself is for cardiac arrhythmias and ischemia. But there's obviously the potential for many more drugs to be developed at specific receptor subtypes. The use of theophylline for asthma was the first clinical use of adenosine antagonists. It blocks the A2B and A3 sites and prevents adenosine's inflammatory and secretory effects in the bronchiolar pathways. The effect of caffeine, on the other hand, is slightly more selective for the A1 and A2A receptors in the brain. In addition to inhibiting the tendency for endogenous adenosine to promote sleep, it blocks the dominant adenosine receptor effect throughout the brain and spinal cord, shown by the relatively larger letter A in this diagram. Some people think that they're not affected by caffeine until they stop ingesting it late in the day. Then they often find that they sleep better without it. Now, just to summarize, in addition to enhancing wakefulness and dilating bronchiolar smooth muscles, methylxanthine acts at all the other adenosine receptors in the rest of the body, but with varying orders of potency. Listed here in the order of their effect, they can dilate renal arteries, contract coronary and cerebral vessels, the basis for their use in migraine in humans, by the way. They can stimulate cardiac muscle. They can also stimulate the medullary respiratory center and improve skeletal muscle contraction and induce emesis. You'll notice that the ability to stimulate respiration is relatively low on the list, indicating that normal control of respiration is relatively insensitive to these drugs. So for this reason, they're not used as respiratory stimulants, that is, analeptics per se. Instead, theophylline is better at blocking the pathological contraction of bronchiolar smooth muscle and the excessive production of mucus during asthmatic conditions, which is higher on this list. Even though methylxanthines have been tested for their ability to improve skeletal muscle function in racehorses, they actually impair their performance because they make the animal nervous and anxious. Very high doses of these compounds will eventually cause a degree of CNS excitation that involves seizures and eventually coma. This may be because at higher doses, they also block the inhibitory effect of adenosine as well as enhance cyclic AMP. You may see panting or vomiting, thirst, diarrhea, and urination preceding the onset of seizures. In the case of theobromine or chocolate-induced toxicity in dogs, death may not occur immediately, but takes sometimes 6 to 24 hours or more to happen. So animals who appear to be excited and are known to have ingested chocolate should be monitored for long periods of time. The order of potency reflects the therapeutic effect of these drugs and the optimal times of their peak effects. The order of toxicity, on the other hand, is actually just the reverse and explains the toxic effect of chocolate with theobromine in it in dogs relative to caffeine and theophylline. A good medical history is important because methylxanthines affect and are affected by other drugs. First, methylxanthines are metabolized by the liver, and many drugs that compete for these metabolic enzymes prolong the effect of methylxanthines, like cimetidine, erythromycin, allopurinol, thiobendazole. On the other hand, if the animal has been previously treated with high doses of phenobarbital, for example for an epileptic condition, the effect of methylxanthines may be shortened. This is because drugs like phenobarbital induce liver enzymes. Finally, theophylline may enhance arrhythmias that tend to be induced by halothane or ketamine. So a careful pre-surgical workup should identify this in asthmatic patients. So how do you treat a dog who's eaten a whole bowl of chocolate at Christmas with theobromine or a bottle of no-dose with caffeine? It's always important to try to remove as much of the toxic compound as possible. In cases of ingestion of the compound, you need to empty the stomach, either by inducing emesis or by gastric lavage. Remember that emesis in cats is best induced by a low dose of xylazine, whereas emesis in dogs is usually induced by apomorphine. In addition, you may need to address the effects of methylxanthines throughout the body. Always support the cardiovascular system when that's compromised. So fluid therapy would be indicated to support a low blood pressure, or use lidocaine or procainamide to treat tachycardia, and propranolol to treat arrhythmias. Respiration might even be inhibited rather than stimulated at high doses, so be prepared to support respiration for relatively long periods of time. There may also be a need for oxygen therapy. Finally, if seizures develop, treat these with anticonvulsants. It usually takes extremely high doses of methylxanthines to induce seizures, but there's a high biologic variability. Some dogs eat chocolate with impunity, while others appear to be much more sensitive to their effects. It's safest to monitor any dog who's known to have ingested these compounds. So methylxanthines have a high therapeutic ratio. That is, there's a high LD50 divided by a small ED50. Small doses are stimulant, probably by one mechanism, and higher doses gradually recruit additional effects that contribute more to their convulsant activity. Now we want to focus on a different compound, doxapram, available as Dopram V. This drug is useful as a respiratory stimulant or an analeptic compound. Analeptics are used to overcome the respiratory depression produced by a wide variety of compounds like barbiturates, opiates, xylazine, or tilazol. Doxapram activates the carotid receptors in the periphery. This produces a signal from the periphery in the circulation that there's insufficient respiration. The respiratory center in the medulla then tells the nerves controlling respiratory excursions to breathe deeper and more frequently. But the analeptic effect of doxapram lasts for only about 5 to 10 minutes. This summarizes the control of respiration. In response to blood gases at chemoreceptors in the heart, high concentrations of CO2 and low concentrations of O2 or oxygen stimulate these chemoreceptors. This triggers signals to go to the respiratory center in the medulla to increase the rate and depth of respiratory excursions. Other things also modulate respiration, but the default system are those two areas. For example, when you're asleep and your respiration is on automatic. The other modulators of respiration include the cortex, the lungs, the muscles, and pain receptors. The cortex provides conscious or voluntary control, so you can hold your breath. Input from the lungs, muscle, and nociceptors, or pain signals, are giving you cognition of extraordinary events, like exercise or fight or flight responses that might require additional oxygen. Doxapram not only stimulates respiration, it also increases body temperature. That is, it causes hyperthermia. And it increases sympathetic activation of the adrenal, increasing epinephrine and glucocorticoid release. It increases cardiac activity, increasing heart rate and blood pressure, and it also increases salivation. These are all important changes in light of the fact that you're probably using this compound when the animal is recovering from an anesthesia, such as after surgery. You want to be sure that the increase in sympathetic tone is not so dramatic that you induce bleeding, for example. Also, the increase in salivation should be monitored to ensure that the airway remains clear. Because the effect of doxapram is so short and the window for its safe dose is quite small, it's usually delivered IV. The rapid dissipation of its analeptic effect means that it can't be counted on to support respiration for long periods of time. Rather, it's usually used to enhance respiration for the short time that it takes to help the animal blow off a gas or overcome a temporary and critical respiratory depression. Doxapram has stimulant properties for many systems, in addition to the respiratory system. Only slightly higher doses than what's necessary for its analeptic effect will also cause skeletal muscle hyperactivity, alkalosis, nausea, vomiting, as well as salivation in the anesthetized animal. So if given to an unanesthetized animal, or if the animal wakes up, it may also cause aggression, catalepsy, and catatonic rigidity. Doxapram can mask the effects of centrally acting skeletal muscle relaxants, but not those of peripherally acting agents like curare or succinylcholine. In other words, if the excitation of skeletal muscle by acetylcholine is blocked peripherally by, for example, curare, you can't overcome that blockade simply by sending more messages to the intercostal muscles. Because doxapram induces the release of epinephrine, doxapram may lead to arrhythmias, especially when coupled to halogenated anesthetics that have a tendency to be arrhythmogenic anyway. The increase in blood pressure will be additive to any increase already induced by, for example, an injection of epinephrine or other alpha adrenergic receptor agonist during the surgery. So be sure to monitor animals continuously after delivery of doxapram to be sure that the effect on other systems isn't problematic, and to know when the short respiratory stimulant effect wears off in case you need to use artificial respiration. Dogs like to sniff things. Their sense of smell is an important part of their exploratory behavior. So it's easy to understand that your pets might think that if it's good for you, then they might also want to sniff it too, just to see what it is. So cocaine and amphetamines are both sources of toxicity in veterinary medicine. They produce arousal, euphoria based on human reports, involuntary motor activity, increased blood pressure and heart rate, and mydriasis, a dilated pupil. If you're going into research or want to get a great salary working for a drug company, it's good to know that as the effect of amphetamine wears off, there's often a reflexive depression and paranoia. It's sufficiently well documented and reproducible that it's even used as a model of depression in rodents to test antidepressant drugs designed for human use. Although your primary interest may not be humans, you should be aware of the symptoms associated with these drugs in the personnel working at your clinic. You should be able to detect possible signs of drug abuse. Often people who use one recreational drug will substitute another that's available in a pinch. You're responsible for the drugs in your clinic, so you need to be able to detect security threats. Drug abuse is actually more prevalent in professions connected to all areas of medicine than even in the general population. This has to do primarily with access. In addition, there's some degree of trivialization of the adverse effects of drugs when you're around them all the time. How do these drugs work? In the brain, cocaine and amphetamines act by inhibiting the reuptake of dopamine, norepinephrine, and serotonin back into their synaptic terminals. This allows these neurotransmitters to interact with their receptors in the synaptic cleft longer than usual. The inhibition of reuptake differs from that produced by reserpine. Amphetamines and cocaine block reuptake from the synaptic cleft area, whereas reserpine blocks reuptake into the storage vesicles inside the synapse. So reserpine depletes monoamines like dopamine and norepinephrine, causing depression, whereas stimulants enhance and prolong the action of monoamines, causing excitation. Cocaine, but not amphetamine, also has that local anesthetic effect on nerves. But the distribution of cocaine is usually lower in the brain than in the periphery when given peripherally, so local anesthesia is usually only seen at the site of delivery. Amphetamines also stimulate alpha-1 adrenergic receptors directly, in addition to their blockade of reuptake of monoamines, making them very potent adrenergic agonists. When cocaine reaches toxic doses, the signs of toxicity include cardiovascular, respiratory, and central nervous system signs. In the cardiovascular system, there are usually arrhythmias, possible myocardial ischemia, myocarditis, and aortic dissection. In this regard, they potentiate the effect of any other sympathomimetic compound. Respiration is usually depressed at toxic doses rather than increased. And there may be cerebral vasoconstriction and seizures. In the case of a pregnant animal, it may precipitate premature labor. In any animal, it can cause hepatic and renal failure. And chronic use usually leads to sloughing of the corneal and nasal epithelium. Cocaine and amphetamines are frequently snorted. Because the concentration is highest at the site of delivery, that provides another clue to potential drug abuse. There's a prolonged vasoconstriction on the nasal septum in the case of cocaine, and this can become necrotic after prolonged use of the drug. So a red and inflamed nose and or widely dilated pupils, mydriasis, may reflect a serious drug problem. Although we've grouped these two compounds together for the sake of discussion, and because of their similar effects, there are a few things that differ between these two compounds. Amphetamines have a longer half-life than cocaine. Amphetamines also have no local anesthetic effect. Amphetamines are metabolized in the liver by P450 enzymes, while cocaine is metabolized by esterases. Amphetamines may lead to anorexia. This is the rationale for the old-fashioned diet pills. However, tolerance develops very rapidly to this effect. If amphetamines are specifically identified as the toxin, the animal can be treated by acidification of the urine. This enhances the ionization of the drug in the urine and promotes its excretion rather than reabsorption. In addition, supportive measures for the cardiovascular, respiratory, and CNS effects should be provided. A related compound is methylphenidate, or Ritalin. Although it calms individuals with ADHD, they're related to amphetamine and have a similar set of stimulant and toxic reactions in normal mammals. The last compound in this category of stimulant compounds is strychnine. It's a rodenticide that produces a very characteristic seizure. While many compounds cause seizures, they usually elicit some form of a clonic and or tonic action. In contrast, after strychnine poisoning, the animal presents with extensor rigidity. This presentation resembles tetanus because the same pathway in the spinal cord is impacted by both strychnine and tetanus. Dogs are much more sensitive to strychnine than cats or humans. Cows and horses would be the least sensitive. This slide illustrates the extended position that the limbs assume with an arching of the back and neck. The drug essentially removes all the normal inhibitory tone in the ventral horn of the spinal cord. As a result, the stronger anti-gravity muscles are allowed to dominate. In addition, you usually see hyperthermia, acidosis, and hypoxia. Hypoxia occurs because the skeletal muscles responsible for respiratory excursions are in a spasm, so respiration is blocked as well. While this is happening, the animal is conscious and aware of the situation, so it's bound to be anxious and panicky and potentially in pain from the muscle contracture. How does strychnine produce these effects? Strychnine inhibits the glycine receptors located on spinal motor neurons. These sites normally serve as feedback inhibitors to motor neurons. When they're stimulated, motor neurons release acetylcholine from their synapses. Peripherally, this acetylcholine is spritzed onto the skeletal muscle at the neuromuscular junction and induces a contraction. But at the same time, it also releases acetylcholine from axon collaterals in the spinal cord. These collaterals project to and synapse on Renshaw cells. There, the release of acetylcholine activates the Renshaw cells, and they, in turn, release glycine back onto the same motor neuron that initiated the whole cycle, as well as surrounding motor neurons. Since glycine hyperpolarizes the motor neuron membrane, it tends to inhibit the motor neuron from firing again for a period of time. This feedback inhibition prevents the motor neurons from firing again too easily. Instead of a tonic inhibition, it provides an as-needed inhibition. The effect of strychnine is to block the effect of glycine at its receptor. As a result, there's excessive activation of skeletal muscles, and the strongest ones, the anti-gravity muscles, dominate. Now, interestingly, in the sloth, this is just the opposite, as they tend to spend more time hanging from trees. Glycine is a relatively primitive neurotransmitter. The reason that the spinal cord is primarily affected while the brain isn't is because there's little or no glycinergic transmission in the newer, more sophisticated brain. But the regulation of motor activity by glycine in the spinal cord has been so successful that it's preserved in the evolutionary development of most animals. The absence of glycine receptors in the respiratory centers in the medulla explains why strychnine can't stimulate respiration. The tonic contracture of the intercostal skeletal muscles explains why strychnine actually inhibits respiration. A clinical note to remember is that tetanus also causes seizures that present with a similar extensor rigidity. Instead of blocking the glycine receptor, tetanus blocks glycine release from the Renshaw cell. Unless treated, strychnine toxicity usually causes death because of the interference with respiratory excursions during seizures. It's always important to remove as much of a poison as possible. In this case, it can be done either by gastric lavage or by neutralization of the alkaloid with, for example, tannic acid or potassium permanganate. If the strychnine is already in the late stage, the poison will have already been absorbed or passed on beyond the stomach. In all cases, early and late, first treat the seizures and support the respiration. An IV injection of diazepam, an anticonvulsant, or an anesthetic dose of pentobarbital will control the seizures. Thereafter, compounds like methylcarbamol can be used to help treat the muscle activity. So why can't you just inject glycine intrathecally to compete with the effect of strychnine at its receptor? We showed in a study published in 1988 that a spinal injection of glycine actually increases seizures induced by strychnine. This is because glycine also has a positive modulatory effect on NMDA activity, an excitatory receptor. So glycine's ability to promote excitatory activity via the NMDA receptor is in itself convulsant. Although there may be many compounds that can induce seizures as part of their toxic effects, these 4 compounds illustrate how differences in their receptors and differences in the location of those receptors in the brain can explain why some compounds are used therapeutically as analeptics to enhance respiration, like doxapram, and others are used for asthma, like theophylline, while others are simply convulsant, like strychnine. The next lecture will discuss drugs used to treat seizures induced by compounds like these, as well as seizures induced by epileptic conditions.